Now, the answer to this can be a little tricky and even somewhat debatable, but our answer here is that the transcription factor would bind less tightly. Now, it probably wouldn't bind more tightly in this scenario because we set it up as the protein binds this sequence really well, and then we change that sequence. So we wouldn't expect the protein to be able to, at the very least, bind as well. It is possible in some scenarios that we lose the ability to bind altogether. But in general, any given sequence of DNA, one protein is making contact at multiple points, at multiple letters. So any given sequence, there are multiple important points of contact. And changing just one of them won't completely obliterate the ability of the protein to bind, but you will diminish its ability to bind tightly. So if we deleted this whole sequence altogether, at that point we're looking at something where the transcription factor wouldn't bind at all. Now, as is true for virtually everything in biology, there are exceptions to every rule, and there may be a scenario out there where you delete one letter and the whole protein can't bind anymore. And we'll even talk about different scenarios at some point where one change in the letter might actually result in a higher affinity binding, the protein binding more tightly. But for right now, our basic purposes, we just wanted to emphasize the point that proteins bind specific DNA sequences, and when we change those sequences or when they're different or not optimal, that the protein usually binds less tightly. There's less of an affinity because we're talking about chemistry here.